Welcome to the PharmaSource podcast. I'm joined in the podcast today by Kerstin Papenfuss, Director of Pharma at Deep Science Ventures. Kerstin is a venture builder working with entrepreneurial scientists to build startups from scratch. And as Director of Pharma at Deep Science Ventures, she's led the creation and development of novel therapeutics meeting unmet medical needs and has created around 50 startups in total, most of which she says are still around. Kerstin, welcome to the podcast today.
Thanks for having me. I wouldn't say I've created 50. DSV in total has created 50, and 15 of those are in the pharma sector that I can actually claim credit to.
Okay, well, 15 startups is no mean feat.
Exactly. And except for one, they're all alive. So
Fantastic. So before we jump into that, I think as an introduction, it'd be great to learn a bit more about your journey, the blend of research that you have with business expertise from Imperial College to where you're at now. It'd be great to understand your journey.
Yeah. So I think it makes sense to kind of start with my PhD, which I actually started at the German Cancer Research Center in Heidelberg. But then halfway through, my professor got offered a full professorship at Imperial. And then basically was the choice between staying in Heidelberg and looking for a new PhD or moving to London. And I've never been a fan of small cities. So for me, moving to London was actually a good thing. We ended up moving the whole lab to Imperial College in London. And that's where I finished my PhD in Tumour Immunology, which these days is very handy because that's where a lot of the action is.
Yeah.
Then I stuck around a little bit just to finish things off and do a few kind of siRNA screens looking for novel combinations in the space of immunotherapies. And yeah, I got quite frustrated because every time I showed them to medicinal chemists, they told me this will never be a drug. And that just told me that I'm kind of missing the trick on all the stars that need to line up for something that is a good scientific finding to something that eventually will have patient impact, right? And patient impact is my ultimate motivator. So that needs to be the focus of what I do. That led me at the time to join LifeArc and help build out their due diligence team. Initially, that was for collaborative drug discovery projects only in the small molecule and antibody space, really looking for early stage ideas for novel small molecule and antibody drugs in the academic realm. That was globally. And then using the drug discovery skills that LifeArc had built up to de-risk those opportunities and then turn them into either licensing opportunities with pharma or into startups. And I did learn a lot during that time. I think in my time there, the remit of the team also grew. So we got involved in diagnostics as well. We got involved in managing pre-competitive alliances with big pharma, so really got that understanding what big pharma is really looking for. So the Dementia Consortium, for example, but also working with like-minded players such as Cancer Research UK, now called Horizon, working together with them in immuno-oncology. So got a really good breadth of indications that I got to work on. So really understanding the state of the art, where we are in kind of solving these indications, but also really good foundation in drug discovery and early stage drug development, basically. And around that time, LifeArc was also very, lucky in a sense that they had all the royalty income from Keytruda. So they started setting up investment vehicles. So they started setting up the venture fund and the seed fund and stuff like that. And my role was, first of all, helping get these due diligence processes off the ground. But also they asked me to set up a scientific horizon scanning strategy to really understand what's the next big thing in science. And at that time, these days we laugh about these attempts, but I did something that was kind of trying to combine basically good early indicators for good science using kind of sentiment analysis from the web, early use of social media data and stuff like that to identify the next big thing early on. And that was working to some extent.
So what would that tell you? Presumably you'd be looking at scraping, I don't know, Facebook, Twitter, whatever. Would it tell you where people are mostly complaining about the afflictions?
No, that was mostly like if there was a groundbreaking scientific publication such as the next CRISPR, people would talk about it. You
Ah,
couldn't
okay.
miss.
So it's analyzing what the scientific community are talking about.
Yeah, exactly.
Mm.
So yeah, well, there was also a lot of like papers talking about the revival of the dodo and the woolly mammoth at the time that we did get. So we had to ignore those. But that basically told me that the future of drug discovery will go into the cell and gene therapy space, or it will go into the AI-informed drug discovery space.
Mhm.
And I got really excited about both. And that was around the same time when clinical trials with CAR T and gene therapies were making these massive headways, right? You could all of a sudden cure leukemia with CAR T. You could make kids walk that otherwise would have died with gene therapy. So I was
Mm.
like, okay, there is a way that we can actually leverage biology to beat biology. And that really fascinated me. Unfortunately, back in the day, that wasn't where life was heading. So I was like, okay, if I want to be part of this revolution in biology, I need to change. So I ended up joining the cell and gene therapy catapult to drive their research commercialization strategy, really. So that was really my role, to work with academics in the UK to basically turn their novel ideas for cell and gene therapies into investable propositions and use the skills of the catapult to de-risk, basically. And on the other side, to really build up a investor network of people who wanted to invest in such propositions, right? So that
Mm.
was like the heyday of cell therapies. And yeah, I found a couple of projects that were academic projects that are now big companies. For example, PureSpring is a good example. But what I most often found was maybe somebody with a good idea for a gene therapy, but then academia is driven by novelty and stuff like that. So it was very focused on the novel biology, but maybe the things that you needed to consider to turn this into a therapy kind of took a back stage, kind of.
Backseat.
Backseat, exactly.
Yeah.
And yeah, so maybe the vector was from the guy who worked across the floor because they had made that before and it works, kind of thing,
Mm.
right? And no consideration to scale up or the actual, how would you use that in a therapeutic setting? Can you actually inject it that way? Would the health economics for something like that really work out? How
Mm.
cheap would you need to do this? Can you actually manufacture this? So I was sitting on this project of things where the underlying idea was good, but the implementation wasn't the best. And that way you end up with suboptimal propositions. And you can't go to
Mm.
venture and say, you know what, the underlying idea is good, but I know we've implemented it the wrong way, so give me money to do it all over. That's unfortunately not how it works. And that was really, really frustrating to me. And I kind of went in my head, wouldn't it be nice if there was this place where you could think about everything from the get-go rather than, trying to jump on a running train and then correct.
Mm.
Um,
Mm.
and that was like my main frustration. The second frustration was that most often I didn't have the most inspiring conversations with a gray-haired PI that is always on the slide deck, but it was actually with the postdocs who had moved from one field and then tried to move into another field and take their learnings with them and apply their knowledge kind of cross-disciplinary. And
Mhm.
those were the discussions that I really, really loved. But at that point in the UK, there wasn't this founder-led biotech that was already thriving in the US. There wasn't anywhere where these people, unless they were working in really highly entrepreneurial labs, could take their ideas forward. And
Mm.
that was like my second frustration. So I was kind of looking for an outlet, I guess, to address those frustrations. And that's when I bumped again into Mark and Dom, who had just started DSV the year before. And yeah, they were telling me about this first principles driven way of building ventures that actually does that, right? It starts with the outcome that you're looking to address or the problem that you're looking to solve, and
Mhm.
then builds the solutions completely backwards to think about everything from the start.
Just to pause on that
Yeah.
first principles, that reverse engineering approach. You've just been talking then about these suboptimum propositions. So you've got
Yeah.
academia that's driven by novelty, not considering how they scale up. And when they finally get there, realize that the market just isn't there. The economics doesn't work.
Yeah.
How do you solve for that? Is there a framework for making that better?
Yeah, absolutely. So what we basically look at, let's say we take an indication and we work out exactly what the state of the art is in this indication. And also
Mm,
not just at this point in time, we also try and predict what it will be in a few years' time when this solution will become relevant and what sort of metrics we would need to hit in order to be able to compete um, with that in that market,
mm,
basically. And also, what I love to do is work on solutions that are actually curative. So that then fundamentally changes the math of the health economics that you're working with. So we're
mhm,
trying to get to a point really early onwards where we're saying, okay, if we can achieve a cure, and for me, a cure isn't something that's like a once in a lifetime treatment, but something that is just applied very rarely, right? Because at that point in time where we are right now, we can't talk about cures because we don't know how to measure them.
mm.
But something that has a much longer patient benefits. That's how I'd like to think about things, and things that mechanistically would make sense in terms of providing a cure, right? So, for
Mm.
example, a TNF antibody would never be a cure just because they just kind of knock the edge of the inflammation, right? But if I have something that mechanistically addresses the cause of the disease, that at least has the chance to be a cure.
Mhm.
And then I just need to find a way of modulating that specific factor in a lasting sense. So we do try and get a feel for, okay, what is tolerable in this market? You know, is it a market that can actually tolerate high price tags that come along with gene therapy delivery vehicles such as Lenti or AAV? Or do we need to think about this fundamentally differently? Do I need, for the way that I want to modify it, do I need to have repeat administration or not?
Mhm.
Or let's say for neurodegeneration, would people actually be open to administration intrathecally or not? So just kind of working through that.
So you're doing all of that work before you even engage potentially with the academic?
Yeah. So we try and get a feel of what the potential
Mm,
solution looks like so that we can then pick the right technology that operates within the constraints. So we call this constraints. So we say a field is constrained by health economics. A field is constrained by who will pay for it.
mm,
A field is constrained by the technology edge, by IP. And we try and map this out pretty broadly and then try and develop solutions within these. And then we will look at each constraint individually, how we could build something that works with that constraint. And if you then have something that works across all of your constraints, then you have
mm.
an ideal solution there. Usually that doesn't happen, so you got to compromise along the way. But the closer you'll get, the closer you are to a solution that won't be leapfrogged by anybody else, basically.
Interesting. And what's the reaction typically then from the scientists to this type of approach? Because it feels like it's quite different to what other conversations they're having, probably.
So it depends. The scientists that we look for ought to be very good at first principles thinking because that's what we do with them. So they help us develop the whole proposal, right?
Mm,
So we pick the ones that do first principles thinking naturally or can understand it to some extent. And they then end up joining DSV on a like full-time basis. So they leave their job behind to then start building out that startup. So they are the people that get it. But often we establish academic collaborations with people who bring components of the technology.
mm,
And oftentimes they are really good conversations because the way we approach them is usually bringing a novel angle to what they were doing anyway. But they hadn't thought of this application, for example.
mm.
So it's a very different conversation to your usual tech transfer model, where when you approach somebody, they always have the feeling you want to run away with their
Yeah,
discoveries. In this case, you're kind of pushing through open doors because you're actually bringing something.
yeah. Do you also then advise them on what happens in between those 2 points in time, like how to build a team and how to take it to the next stage? And
Yeah, absolutely. So once we have the first, we call them founder in residence. So let's talk you through the process a little bit. So once we have
mm,
a problem that we're looking to solve or an outcome that we want to achieve, then my in-house team will kind of map out what this could look like most likely. and where we're going to find the most likely success. And that'll tell us what sort of expertise we are looking for in a founder in residence that then helps us build this all the way to the end. And that founder in residence then closely works with the internal team to build out various solutions, right? There isn't just one way of solving a problem. There's usually many. And then we try and whittle down the best way. And once we have an understanding of that, that usually requires bringing in complementary expertise. And then we help with the recruitment of that complementary co-founder, right? So we always look out for technical expertise, but we're also looking for people that will thrive in the startup environment, for example, right? So being able to deal with uncertainty, being resilient, being kind of magnetic, because you need to be able to raise the money, you need to be able to convince people to join your team. So those are the types of criteria that we look out for in the founding team. And once we have the founding team, then we try and develop everything. So a proof of concept plan for the idea that we're looking to basically validate, which is always orchestrated together with the IP strategy. So whilst our companies don't start with proprietary IP, from the get-go, they always have plans to get there within the first few months, basically, to get to a position where they can file for IP. And that's then usually tied in with a fundraising strategy where you have the value inflection points at the right stages so that you can, do everything in tune, ideally. And then we also help with bringing in advisors that then can make that team look very credible in front of investors, and then provide the pre-seed money to get this off the ground, basically.
It'd be good to understand a bit more about something you've described as being your problem idea spinning process. How do you go about identifying the ideas which are worth pursuing?
So there's kind of multiple steps that we walk through. So we started with a problem usually, and there we kind of whittle it down based on downside, upside, and neglect, right? Downside is what sort of problem is it, and how many people are looking to solve it. Upside is what would happen if you were to solve it. Could you make enough money? And there is
what would you say those attractive problems are right now?
Obviously, there's still lots of indications where we have no working treatments, right? You could start with neurodegeneration, right? I don't believe for a second that A-beta antibodies will solve the whole puzzle. So you could think about novel therapeutic approaches, but you could also talk about the enabling space quite a lot. And I think that's where I am quite excited right now, because I believe in that we have the biology that we can build curative approaches. We don't have the right tools yet, right? So anything that broadly unlocks a therapeutic modality would be something that I'd be really, really keen to kind of consider. And just kind of give you an example that I'm going to talk about later, which is the company called Plurify that we started with the Cell and Gene Therapy Catapult, which was really focused on that problem that one thing that will stand in the way of scaling cell therapies is actually, can we make a product that's pure enough to put into patients? And none of the existing methodologies, for example, work. So this is a problem that everybody who's developing cell therapies to a point where they need to scale will run into. So it's a massive problem, right? So those are the types of problems that I get quite excited about.
Interesting. You develop those problems into ideas.
Exactly. So basically, we rank by downside, neglect, and upside to understand which of the problems are basically worth solving or really, really good. And there is kind of traction in the field right now. So, for example, female health is a massive space of neglect, right? Not enough has been happening there. And then kind of have a look at the spaces where we can actually deploy the DSV methodology. So DSV's methodology works well in fields where there's lots of data availability, and you can see stuff that has worked, hasn't worked, and then kind of can hypothesize why things haven't worked, and you can come up with novel ideas on how to fix things. It doesn't work well in spaces where there is just no data. White spaces, it doesn't work, basically, right? Because you can't engineer on the back of no data. So once we have an understanding of that, we kind of combine the two and then home in on these problems. And then we take them really apart based on a first principles level. So we're saying, okay, this is the problem. can we ask first principles questions on how to unlock the constraints? So we start mapping out the constraints that I've talked about, and then ask first principles questions on how to break that constraint. And usually as soon as you manage to break or flip a constraint, that will basically unlock a whole new solution that nobody has ever thought about, basically.
How do you flip a constraint?
Yeah, for example, let's think about how we build what's now called Deliver Bio, right? So this is a company that are now doing in vivo CAR T, right? But we created this back in 2017, and that's where people had realized that the autologous manufacturing process probably was going to be too expensive to really scale into mainstream applications, and everybody was saying, can we do this in an allogeneic way? And that's where we saw most of the tech development, right? But they asked the first principles questions that said, okay, everybody is operating under the assumption that you need to do the manufacturing outside of the body. Can you actually do this inside of the body? Can you break this assumption? Does it have to be outside? What happens if you were to achieve to do this inside the body, right? And if you were to do that inside the body, that then moves this from basically a cell therapy approach into a gene therapy approach, and something that should be available off the shelf straight away, which basically would cut down manufacturing costs massively. And that way you could be able to turn this into a modality that can break into the mainstream market.
You mentioned Plurify earlier. What was the story of Plurify, and how does it apply your first principles to cell therapy development?
So this is the company that we built together with the Cell and Gene Therapy Catapult, which was a fantastic collaboration because the Cell and Gene Therapy Catapult obviously has this very advanced understanding of the cell and gene therapy space. They are ahead of what the industry should be doing, so they should be aware of all the problems, right? And with them, we homed in on this problem of purity, really. So we basically said, so for any regenerative cell therapies to become scalable, they need to be based on a stem cell background, and there could be embryonic stem cells or iPS, which is obviously the best solution, probably. But for these approaches to work, you need this high degree of purity because you don't want to be putting stem cells into patients. Even if they are in a tiny amount, they have the massive cancer risk. And that's what we were even observing in the build process. There were publications coming out where people had been doing this for diabetes, and then the patients had cancer, basically, right? So a massive problem that will hold all these solutions back until you fix it. And then that's obviously the safety risk, but it also does come with a efficacy risk. So you start with the stem cell that you're trying to differentiate into your cell type of choice that you want to put into the patients, right? But we haven't fully understood on how to drive these processes in the most efficient way. So sometimes there is other cell types in there that you don't actually want because they might impact your efficacy, for example, or you might just be wanting to be able to control how much of a mix you have because you need to have a reproducible mix to be able to put this into patients, right? And at that point, everybody was using either cell sorting through like FACS or microfluidics, right? Or even magnetic sorting to sort their cells that they want to put into the body. And that works for small scale clinical trials, but it doesn't work for your large scale clinical trials in indications such as cardio, where you would need 10 to the power of 13 cells, right? You can't put them through a microfluidics device. I mean, it's micro, right? So that wouldn't work. So we were like, okay, we need to find a scalable solution. And then we started defining the constraints. So scalability was one constraint. Another constraint was that whatever we come up with doesn't leave a trace in the end product, right? Because we don't want to mess with CMC. We don't want to leave anything behind. Things like CRISPR and everything needs to be done in safe harbors. We're like, okay, let's not even go there. Let's just not leave a trace. We need to have something that can be basically plugged into any manufacturing process because people are doing this in various different ways, and we need to have something that's compatible. Those are the sorts of constraints that we set from the outset. And then we were asking first principles questions. What are other ways of doing this, basically? And basically, somebody around the table was saying, wouldn't it be nice if the cells realize themselves that they are the wrong cell type?
Right. Okay.
And that would be nice, wouldn't it?
That would be
Right?
nice. Yeah.
That would be ace. And we're like, okay, can we teach the cells to understand what cell type they are? And can we impart this capability? And that's kind of what started this thought process. I can't talk about in detail of what it ended up because these guys are just, well, they have filed their first IP, but I don't want to talk too openly about it. But basically, it's a neat tech bio solution that exploits biomarkers together with a modality that doesn't hang around to really drive the purity of the cells. So basically, the cells that are unwanted can be selectively eliminated in a way that's completely scalable, right?
How interesting.
And that solves a major industry bottleneck, and we're already seeing that in the types of traction that that company gets. So they're getting a lot of inbounds from the people who are actually running into these problems.
Let's talk then about the way that different partners might interact with you and also with the startups that you work with. Obviously, you work with big pharma, you work with disease charities,
Yeah,
ecosystem
yeah.
partners. How do they complement each other? What's your take on how it all fits together?
Yeah. So we love working on problems with people who have an in-depth understanding of that problem, and that's kind of how we pick our partners usually.
Mm,
So as you say, they could be disease therapies because they're obviously expert in that indication. I mentioned the cell and gene therapy catapult because they have this in-depth view. Could be corporate partners who want to do something in a specific strategic area, just area where they have a lot of capabilities but don't see the deal flow, kind of, right? And what we like them to bring, other than obviously co-financing the build of that company, we like them to bring that, deep subject matter expertise. We at DSV, we are generalists. We have a really good understanding of the edge of biotech as a whole, but we are not detailed experts in that specific problem. We just like to ask really good questions.
mm.
So we are looking to the partner to bring that sort of expertise or access to that sort of expertise, right? So these disease charities, for example, are super useful because they have the whole network of academics that are the key opinion leaders in the fields. And for me, it's much easier to get one of them on the phone if the charity does the introduction for me and they actually do pick up the phone, as where I were to reach out cold and they're like, who is Kerstin from TSV, right? So that really for us speeds up the whole process of moving from idea to something that actually has legs because these people take our calls and respond to whatever we want to run by them, right? And that's really enabled through the partner a lot of the time. Then often people sit on data sets that they don't know what to do with because they don't necessarily have the skills in-house. So that's also an expertise that we bring in. We have a really good understanding of computation, have our own in-house AI venture creation agent that we have built that can help us dissect those data sets. And yeah, access to talent. So oftentimes, talent is in the networks of these disease charities, for example, that want to start a startup but just don't know how to. So those are the people that we are trying to grab.
Yeah. And presumably the big pharma companies ultimately might well be the ones who would acquire the startups further down the
Yeah,
road.
so they are doing this externally with us because we would be probably going after riskier things and they could shoehorn through the internal funnel. But obviously, if it pans out, then the idea would be that they kind of get first dibs and they get to see the company grow, right?
So when you're working with these various different partners, how does confidentiality fit in? How do you protect individual intellectual property while still creating this more open, innovative
Yeah.
ecosystem?
That is a good question. It is always possible to find a way to communicate without disclosing too much, but still have a lot of learnings. So we do that usually in a staged way. So if we were to reach out to somebody to just get their feedback on something, we try and talk at a level where that doesn't give too much away, but just gives away just enough. And then if we feel that this other person is really interested in what we're doing and helpful for what we're looking to achieve, then we will go ahead and sign that CDA. So we do that quite a bit. But generally, there is lots of learnings that one can actually talk through without disclosing so much. So we are just really careful with how we use our language, basically. It doesn't give away the secret sauce. And I think generally, there's a lot more people kind of wanting to pay it forward out there in the ecosystem than you might think. So you get a lot of like people really willing to help these early stage startups with their expertise, and they are willing to sign these CDAs if necessary. But they're also kind of happy to help with their networks. It is possible, it isn't easy, and you also need to time very carefully when you have these discussions. So, for example, if I'm talking to somebody that has the potential to acquire my company and has all the expertise and capabilities in-house to basically recreate my solutions, I might want to just wait until I chat with them, until I have my ducks in a row and my IP files, right? So it's really kind of a call based on the risk, right? But I think what we've seen across the portfolio is that the people who are a little bit more open and paying it forward are the companies that are actually moving the quickest. Because in the early startup scene, you really depend on this network of people that want to help you. And they can only really help you if they know what you're doing, basically. You know, I'm at an event and I see somebody saying, oh, I can help people do X, Y, Z. Then I will make that connection for a portfolio company and say, okay, you would benefit from talking to each other, right? And there's lots of people that be happy to do something like that for you, but they can only do that when they know what you do, basically.
Yes.
So people in our network that were a little bit too hesitant about talking too broadly, they are the ones that are a little bit more behind, I think.
A lot of what we've been talking about is in the cell and gene space,
Mm.
which, has had its ups and
Yeah.
its highs and its lows. And if you plot it on the Gartner curve at the moment, it's probably somewhere in that trough of disillusionment kind of area.
Yeah. Gartner
Um,
Innovation Core, my favorite thing.
yeah,
Yeah.
yeah, yeah. As a company, how are you feeling at the moment about cell and gene? Where do you see it going over the coming years?
So yeah, I think you're absolutely right. We're in this trough of disillusionment, and we're now waiting for the path of enlightenment to
Mm,
kind of kick in. But what we've seen with cell and gene therapy is not something that's unusual for innovation, right? So in innovation, you always have this. Okay, those are the things that could massively move the needle, and then everybody jumps on it.
mm,
And then these first generation things, they run into problems, right? And then you need to be ready. Um, with a second generation of things that will address these problems that people have run into to build something that would actually work, right? For me, that will happen with cell and gene therapies for sure. We've just seen the first generation, and we all
mm,
know now what the problems are in gene therapies. There are, repeat administration, things not lasting as long, vectors disappearing, things being too expensive.
mm,
So whatever you need to do as a kind of second generation product, you need to be addressing these problems. Now, how long the time is between the trough of disillusionment and the
yeah.
path of enlightenment, I think if I had a way of predicting that, I would be quite rich by now. But you need to start building in the trough of disillusionment so that you're ready when that path of enlightenment kind of kicks in. And in that time, there won't be much cash. And it's also
Mm,
very influenced usually by macroeconomics, right? So we were in this kind of luxurious position like 5 years ago when there was groundbreaking data in CAR-T and gene therapies, and
mm,
that then coincided with COVID that brought in all this money that didn't even know biotech and was basically swamping the whole market. And that money has gone because people have lost their money, basically. But
yep.
it was also not smart money. So now we need to kind of wait it out until this picks up again and we see the first successes. So usually the tide will turn when one of the companies that has survived and is addressing these issues starts to have positive data. So that's
Mm,
usually what triggers the return of a market like that. But
yeah.
as I
Yeah.
said, when that will be, I don't know whether I firmly believe that will happen 100%, right? Because I think we're just kind of scratching the surface of what cell and gene therapies can actually be used for, right?
Mm, mm,
So far we've been kind of treating niche indications, but the way that we are looking at them now, for me, I view cells as a data generation machine, integration machine, basically. You get a signal, the cell receives the signal and triggers an action, right? And at the moment, we've just been exploiting this in the context of CAR T, where it sees a cancer antigen and then it triggers a kill circuit, kind of, right? But there's loads of other functions that we could be triggering, loads of other cell types. And I think with the understanding now that we get through single cell analysis throughout the body in different indications, we will get a much better understanding of what individual cell types do and how we could potentially change them and how we could change those specifically, or how we could use a basically data integration machine such as a cell to change that from the outside. And I think that's fundamentally where this will be going. How long that will take, no clue. But will it happen? I'm sure.
I think everyone's agreed that if you fast forward 20 years, say, the impact of cell and gene will be utterly transformative to humanity.
Yeah.
It's just how long is it going to be like waiting? How much cash do people have to burn while they're waiting for that?
Exactly. How much cash do you have to burn? But I think it's also on us about how we allocate that cash and how much we factor in the cost of development already early on, right? Because I think what we've seen is the cost of these kind of first generation products, which are outrageous. But I think these days you can make things way cheaper than you could already make them 10 years ago. So it's just waiting for this to pan out, and also about integrating this already when you're building your first solution, right? Because if you go down the route with the cell and, well, any therapeutic modality, if you go down the route too far, you can't change it anymore, because then the cost of going back will be too high. So it's really, really essential that you think about this really from the start.
Will cell and gene have its deep seek moment where therapies that are currently at, $1 million per patient can be brought down dramatically? What technology is going to enable that?
I think they will be brought down dramatically. Will that be one solution? I don't think so, because you're fundamentally trying to solve different problems in biology, and there will be things that are much better suited to one solutions than other, right? So I think we will need to have basically an armory of things that can be used to basically, this will be the best modality to solve problem X, this will be the best modality to solve problems Y, and then we will have probably standard technologies, how you get stuff, for example, across the blood-brain barrier, right? At the moment, everybody's kind of tinkering with whatever they think will be best. I think 5 years' time, we will know what's best, and then everybody will be resorting to that. So that will bring in standardization. So that then de-risks your development path as well, because you know how to get stuff in there.
Absolutely, yeah. So moving towards like a standardized armory of different building blocks.
Yeah.
Based on your experience working across various different startups, what advice do you give when you're meeting with entrepreneurial scientists who want to translate their research into something that actually is impactful for patients? What are your sort of go-to advice, best practices?
The first piece of advice, I obviously I've drunk the DSV Kool-Aid, so it's like start with a problem rather than a solution, right? Because it's really, really unlikely that you come up with the best potential solution to a problem just like that, right? Usually you need to start, iterating, and eventually you will come across what you think will work best. So if you have one solution, force yourself to really think about what would be other and different ways of solving this. I think that's quite key, because then you will start factoring all these constraints that I've been talking about. That I think is for me key. Another thing is that people are a lot more collaborative than you think. So the whole DSV model is basically work based on collaboration and networks, right? We don't have any premises, for example, right? We operate mostly virtually, but we do bring people together. And I think you can pull quite a lot together really, really early that looks like a good company without actually having any premises, without having too much while still having quite lean operations. So really use the network that's out there, that is there to help you. And I think that's where we're really fortunate in the UK. You have a lot of things out there to support early stage companies. You have all these accelerators that give away free lab space that come with their networks. You have really good non-dilutive funding, which a lot of our companies actually do get. So DSV companies have a really good track record of getting innovate funding, for example. Um, what else? Yeah, there's a lot of people out there that want to make those startups work. I think we're very fortunate. Me being German by origin, I often ask myself the question whether the DSV model would work in Germany, and I'm not even sure it would, just
really?
because there's a very different risk appetite in Germany. Like quality of living is very, very high. Salary levels are relatively high. And if you were to start a startup and then fail, you'd be seen as a failure, right? I
Okay.
think we have a very different culture here in the UK. Not quite as out there as in the US, but probably somewhere in the middle that actually, celebrates people who try something new.
What about the US? Do you have an office there or any intention to have one?
We don't have an office there because we don't even have an office here, right?
Yeah.
But yeah, we do work a lot with the US, right? This is where a lot of the bigger checks do come from. A lot of our portfolio companies, they get their early stuff done there and then eventually start opening up offices there. Yeah, I have quite a few trips to the US lined up, and I think you can't operate in the biotech market and ignore that.
Kerstin, it's been a pleasure speaking to you today. Thank you so much for your time. Very interesting.
Likewise, you're welcome.